Hi there. In my absence, I figured I'd do this with a sense that I am presenting to all of you from one of me, and I hope you're all having a great day today. This is the present progress for the course MC2201. I guess that was given to me by John Sacco. Microbiology for healthcare professionals. This will also include a lab section. I will give you some details about that. This is targeted for spring 2018. This is the textbook. It's going to be from McGraw-Hill. It is Microbiology, A Human Perspective. This is the 8th edition. Actually, I worked with the 4th edition, Nestor's book, and it was a very, very good tool. It is basically an excellent book that is oriented for healthcare professionals, whether it's nursing or otherwise. There's going to be not only a hard copy that the students are going to have to get, but they can get there's also an online tool called Connect that will be required. And I provide the ISBN for the package. There is an optional learning tool. It is totally up to the students if they want to use it. It has been helpful. You can obtain it from, yes, even Amazon or your local online bookstore. I would not require this, nor would I have the students purchase it and I would teach out of it. I'm not going to do any of that. This is a helpful book if they wish to do this on the side. And basically it's a good one for visual learners particularly. Yes, it is a coloring book. The overview. This course is oriented to the clinical needs of the healthcare professional, and it'll provide students with an introduction of the microbe with the emphasis on the nature and behavior of microorganisms, the interrelationships between microbes and the human host in disease and health, and the principles of prevention, control, and infectious disease. You can see all the various different types of pathogens that I will be reviewing, as well as the various microbial infections, respiratory, digestive, etc. Also, I do take special note of several topics: antibiotic resistance, immunizations, wound infections, and bioterrorism. I've kept that in mind. The students are expected to have successfully already passed Anatomy and Physiology 1 and 2, as well as having a solid basic understanding of biology. Students have basic computer skills, including functional skills in an online learning course, writing using MS Word, and working with an online learning tool such as Connect. If they've had previous experience with mastering A&P, they pretty much are going to be able to do the same thing. I'm going to have it where I will do a review beforehand. At the successful completion of this course, the students will be able to, and I list all the learning objectives. This is also put into the syllabus as well as the prior page. Basically, they're going to be able to discuss the structure function of bacterial cells, identify viral, bacterial, fungal, protozoan, and helmintic pathogens in humans, have the basic concepts of epidemiology and counter strategies to infection, the mechanisms of various actions of various microbiological, antiviral, and antiparasitic medications, discuss the mechanisms of bacterial genetics, metabolism, and gene expression, also understand the structure function of viruses, viroids, and prions, the role of vectors in transmission of viral, bacterial, and protozoan pathogens. Identify the types of helminths and their life cycles. Identify pathogens from infections of the nervous, cardiovascular, alimentary, integumentary, genitourinary, respiratory, and lymphatic systems, as well as infections in wounds and the blood. For the lecture section, I'm going to include narrated lectures with accompanying lecture notes. Also, there will be YouTube videos and articles added to enhance the student content. Also, once a week, I will do a chat. That'll be live using the big blue button. I think Dan told me it was some other name, but anyways, thank you, Dan. This will be set up on Saturday about noon for students' questions and discussion, as well as reminders of upcoming assignments. This will be recorded to help students who are not available for the live chat. This is the section topics, and this is also placed very directly into the syllabus. So the first week we'll be doing an introduction and cell structure review, dynamics and control of microbial growth, metabolism and gene expression, bacterial genetics and biotechnology, diversity and classification of prokaryotic organisms. Now, I'm not going to include some details that are going to be involving mining mining bacteria. So don't sweat that stuff. Most of this stuff is going to be focused on that which would be relevant for the healthcare area. Eukaryotic members of the microbial world, such as protozoans, etc., fungi. This also, there will be a section for viruses, viroids, and prions. The innate and adaptive immune response and applications of the immune responses. So this will include things like vaccines, natural and artificial, basically immunity. And we'll continue with this. Host microbe interactions and epidemiology, antimicrobial medications, food microbiology, skin and respiratory system infections, alimentary and genital urinary system infections, nervous system and wound infections, blood and lymphatic infections. Now, how am I going to do all this? Lots of stuff they're going to have to go through. Lecture section assignments. They're going to have to, of course, view all the videos that I provide for lectures, but they'll have weekly chapter quizzes located in Connect. They will have critical thinking assignments. This is going to be a little bit different here. What I'm going to do is have a pool of about 10 to 12 questions each week, and the computer will randomize and issue them out to the different students. That way, all of them don't have one question, so they can't all cheat off each other. But what the students will need to do is to focus on their own work. There will be a rubric set up. And what these questions will be are, they could be case studies, they could be involving YouTube. They're going to have the question, and they'll be asked to select critical thinking. Basically, they'll be asked to provide the answers to this. Now, these questions are going to be critical thinking, problem solving. They'll need to answer them in 3 to 5 complete sentences and include a bibliographic reference to cite their source. I'll give you an example at the end of this presentation. Now, in the lab section, we're going to be covering the topics of microscopy, aseptic technique and staining, identification of unknown bacteria, isolation methods, microbial growth and control of microbial growth, bacterial genetics, microbiology, and DNA techniques, medical microbiology, an ELISA test that will be a online simulated lab situation. Then we'll be covering viruses and fungi, protozoans, helminths, and vectors. Now, these are going to be animated labs, so the students don't have to have anything, and they're not growing any bugs at home. They're going to be using the mouse to perform actions for each of these lab exercises. Most of them are going to come from the Connect microbiology labs. There are a few that are been set up from the online resource of Howard Hughes Institute, Medical Institute. For example, the ELISA is one, and I'm probably going to use a piece from their viruses demonstration. There are also going to be several labs that include microphotographs for larger scale organisms. That's the vectors, the helminths, protozoa, etc. For those that are not familiar with the term vectors, these would be the disease transmitting organisms. So it could be a flea, it could be a tick, it could be a mite, it could be a mosquito. Everybody knows mosquitoes, and they can transmit all sorts of fun things. Furthermore, there will be some YouTube videos that will be included, especially for the lab for antibiotics. I found some excellent antibiotic-resistant videos that are going to be informative. Some of them are actually going to be from the TED presentations. You probably heard of those great lectures series. Now, the course is based on a 75% and 25% lecture lab breakdown. The lecture grade contributions are going to be weekly assignments, 3 online exams. The questions are taken from a randomized pool of exam questions. One final exam will be comprehensive, and it will be conducted on campus during finals week. The lab grade contributions, you're going to have weekly assignments. These are going to be lab question handouts with discussion questions or problem-solving questions related to the lab they did. And then there will be also 3 online lab exams. This will meet the constructionist requirements. It provides concepts that students learn. They will then reflect to solve the problems and devise solutions to concepts based on what they have learned or what they can determine via investigation, particularly for the critical thinking problems. It meets requirements for the healthcare students, nursing, etc. It makes the students connect the concepts to the healthcare and medical procedures. It meets the social justice topics. It discusses the effects of poverty, poor public health, squalor, civil discord, terrorism, and poor